This is a part 5 of somaclonal variation. Now we are going to various method to detect this somaclonal variation. Now till now we have what we have covered, what is somaclonal variation, how they are produced, what are the causes, and what are the basic factors behind the somaclonal variation. Okay, and now the next topic is how to detect. So now in order to detect the somaclonal variation, first is analysis of morphological character. Quantitative characters such as plant height, maturity date, flowering date, leaf size. Qualitative character, they may be yield of flower, seed, wax content. These are the morphological characters. So very first, the basic what we say reason or the basic very firstly we can observe the somaclonal by variation by using its studying the plant's morphology. In morphology we are studying various qualitative and quantitative character. Qualitative characters are like plant height, maturity date, etc. And in qualitative character we may look for the even yield of flower, seeds, color, etc. Now this variation can be detected by using the cytological studies. So cytological studies are like staining of meristematic tissues like root tip, leaf tip, okay, using various nuclear stains like Fulgen stain, acetocarmine, and moreover we can also go for construction of karyotype, construction of karyotype. So if we study the karyotype of that particular plant, then we can able to detect these changes in the chromosomal number. Then there is a detection by content of DNA. Okay, by using various stains cytometer method, we can detect whether the DNA content has been changed or not compared to the parent. Then there is a gel electrophoresis method. In this gel electrophoresis method, we can isolate the isozymes, and then we can separate these isozymes using a gel, and we can detect the difference in the pattern of isozyme using gel electrophoresis. Then there is one more character, that is detection of disease resistance variant. Okay, pathogen or toxin responsible for the disease resistance can be used to select the agent. So we can apply the artificial selection agent. Okay, we can subject this plant to some disease, and if among these 1,000 plants there may be chances that few plants are resistant to this disease. So that can be, so in this way we can detect the somaclonal variation. Then there is herbicide resistance. Okay, similarly we can subject this plant to herbicide treatment and look if these plants are resistant to herbicide or not. Then there is another method of environmental stress tolerance. So now there are various types of cell environmental stress, just like salt stress, water stress, temperature cells, minimum mineral toxicity stress. So now these tissue culturally produced plants are subjected to the various stress conditions. It is known that naturally plants getting died, but few plants may survive these conditions, and hence we can detect the more beneficial somaclones. Okay, so now we'll discuss what are the advantages of somaclones. Okay, number one, it help in crop improvement. Definitely we can detect the newly improved variety. Then there is a gradation of additional genetic variation. Obviously for breeding purpose two parents should be diverse, and these somaclonals are nothing but the variation. So there is a additional genetic variation has been created. Okay, increase and improved productivity of secondary metabolite. Most plant cell produces various secondary metabolites which has commercial importance, and hence these somaclones may produce improved secondary metabolites. Selection of plant resistance to various toxin, herbicide, high salt concentration, and mineral toxicity, and these are useful for the various breeding program. Now disadvantages. A serious disadvantages occurs in operation which require clonophone uniformity. Now when does this happen? Now we have produced 1,000 plant and we have to distribute this plant to farmer. If farmer plant those plants on his field and then he observes somaclonal variation and that are not beneficial, then there is a problem. Okay, so for industry purpose there is a need of uniformity. Okay, so these somaclonal are disadvantages. Okay, sometimes it lead to undesirable result. Selected variant may are random and genetically unstable. Okay, so it requires extensive study and field trials. Okay, these are the few drawbacks of somaclonal variation. Now, till now, in these 5 lectures, we have clearly covered the somaclonal variation. Now if you have any kind of doubt, you can WhatsApp me. Okay, so this video is particular for the students, and please be prepared. I have also given a separate notes for these particular topics. This is a critical topic, this is an important topic, and there may be a question of 8 marks on this topic, so be prepared. There are very definitions that are hidden in topics, just like caliclones, mericlones, okay, protoclones. So you should be thorough with this. Okay, thank you, and here I conclude my topic, somaclonal variation. Thank you very much.